Hello, everybody, and welcome to our DIME December Journal Club. For folks I may not have met yet, my name's Jen Goldsack. I'm the CEO here at the Digital Medicine Society, or DIME, and I'm absolutely delighted to welcome friend and colleague Visar Berisha to lead our Journal Club discussion today for folks who have had the chance to read his most recent publication in Nature Digital Medicine. We've got an awful lot to learn today about the sort of proliferation of digital data and also the scarcity of high quality data and sort of how we handle all of this. Visar, before I introduce you to everyone properly, let's go in and orient folks to Journal Club if they may not have attended before. So folks, if this is your first, and I've jumped over a few things, if this is your first Journal Club, this is my favorite monthly event. We keep the group smaller. We are inclined towards conversation and questions and discussion. So this is how we'll work. So first of all, I should disclose that today's session is being recorded. The good thing about that means that if you have friends and colleagues who should be listening right now, you will have the recording available. We do post it to the DIME website. They will be able to access both the recording and the slides after the fact. As I said, we do try and make this a sort of high touch discussion based environment. So once Visar has sort of given us a look behind the curtain and given us the author's perspective on this really terrific publication, we'll throw it open to discussion. So we'll stop sharing slides at that point. For folks who are able, we'll go videos on and we'll have a bit of a conversation with Visar, who is joining us today. So without further ado, let's get into our session. So we are going to be looking at digital medicine and the curse of dimensionality today, which first and foremost, Visar, is an absolutely fantastic title. It's one of those ones where I think we need to step our game up over here at DIME. What I'm going to do, I'm going to stop sharing. I'm going to hand over to you. I'm going to let you tell folks on the line a little bit about your expertise, your background, and why we are so lucky to have you leading this discussion today, and then give everyone on the line a little bit of insight from your perspective into the recent manuscript.
Thank you so much. Let me just share my screen here. And thanks for the compliments on the title. My daughter said it sounds like a Harry Potter movie.
It does, and you know, they usually do very well, Visar.
Let's hope that this is as popular. Let's see. Okay, can you see full screen now?
We've got you.
Excellent. All right, thanks everyone. So just a couple of words about myself. I'm a professor at Arizona State University. I actually have a joint appointment both in engineering and in the College of Health Solutions. So my research sort of resides at the intersection of these two interdisciplinary fields. And the work I'll be talking about today started back in 2013, 2014 with my close collaborator, Julie Liss. And there, Julie has worked on speech neuroscience and the manifestation of neurological disease in speech for decades. However, in 2014, we started thinking about how do we actually apply technology to this problem? How do we develop biomarkers based on speech? And there was this formative experience where we developed this model in lab that worked really, really well. We followed best practices for supervised learning. The model seemed to work really well. We were really excited, and they said, let's actually try this out on a clinic on a small set of patients with ALS, and we'll kind of track them over time. And the model failed miserably. And we were really puzzled by this. I mean, we sort of expected that there would be some decline in performance going from lab to in clinic, but we weren't expecting total failure. So that was a formative experience in terms of shaping our research agenda moving forward, shaping the way we thought about the problem. But then I also wanted to provide an explanation for why these models failed. And so while the application area today might be speech, this generally applies to any high dimensional problem in clinical applications. And so hopefully by the end of kind of my preamble here, prior to us getting into the Q&A session, there'll be a good enough foundation where we can have a robust discussion that follows. Okay. All right, so one of the promises of AI, right, is that we can distill these behavioral signals down to clinically relevant information. Things like diagnosis or a measurement of a clinically meaningful aspect of health. But these constructs, diagnosis, clinically, a measurement of clinically meaningful aspect of health, these are high dimensional, complex, heterogeneous constructs themselves. And what's more, the signals that we're acquiring from the human body now are themselves high dimensional. Data from wearables, data from imaging, different imaging modalities. And so on the one hand, that's a good thing, right? Because you have more opportunity to find signal. There's perhaps a higher likelihood of being able to come up with solutions to these really difficult so-called wicked problems. But on the other hand, there's more opportunity to find signal, which is part of the curse, right? Like if you apply AI to a problem and there's any opportunity to find signal, to find separation, it will, regardless of whether it's real or not. And so when you look at the literature, being a consumer of kind of the digital health academic literature, almost every paper discusses the benefits of these new modalities, the benefits of this new high dimensional, high volume, high velocity data. And I thought very few sort of addressed in what I hope is a digestible way some of the challenges associated with working with this data, some of the challenges associated with building models with this data. Okay, so now what are these figures on the slide? Well, what I want to do is to try and demonstrate some of these problems using geometric intuition, and again, using an example from the speech-based biomarkers literature. I want to consider this really simple problem, simple to state, really difficult to solve. So the simple problem is we want to capture speech from an individual and build a classifier to determine whether the individual is healthy or has some form of mild cognitive impairment. And we're going to consider sort of two versions of reality. In the first version of reality, in the first instantiation of this problem, there's only a single feature that we've extracted from the speech data that is relevant for solving this problem, this so-called type to token ratio. All of the samples here represent data from different patients. So the blue patients with MCI, the red are healthy controls. And as you can see, in this one dimensional feature set where we have a single feature, this type to token ratio that varies between 0 to 1 in my notional example, we have a pretty dense sampling of the feature space. We have some patients or some participants with low type to token ratio, some participants with high type to token ratio. And it seems like we've sampled the space rather densely, right? Now, let's consider a second version of this problem. The second version is now we have actually two features that are relevant. The first is the type to token ratio on this x-axis here, but now we also have a second feature, this lexical density. So if we now, on this two-dimensional feature set or two-dimensional feature space, plot data from the same participants, you see that the data is much more sparsely situated. So the distances between points is greater. And you don't have a very dense sampling of this two-dimensional feature set because effectively the complexity of the problem has increased. You have now a two-dimensional problem instead of a one-dimensional problem, but the sample size has remained the same. And so what are the consequences of this? One of the concepts that we introduced in this paper is just this idea of a data set blind spot. These regions, these contiguous regions of feature space are what we call, that are colored in green here on the two plots, are what we call data sets blind spots. Those are simply regions of feature space for which we don't have observations. So we have no idea what's happening in that region. And as you increase the complexity of the problem, so as you go from one feature to two features to three features, the volume of this data set blind spot grows exponentially. So you can see the blind spot is much greater in 2 dimensions than it is in 1 dimension. And this is by virtue of the fact that you have the same number of samples, however, they're farther apart. And so now there's bigger regions in feature space where you're effectively undersampled. So the volume of this blind spot increases exponentially with increasing problem dimension. And practically, that means that there are more combinations of features for which you have no idea what's happening. You don't know whether individuals in that region belong to the healthy class or the MCI class in our example. Okay. So how big of a problem is the blind spot? Well, that depends on whether it's important or not, right? So let's in the paper, we kind of talk about different reasons why blind spots come up. I'll very briefly touch on a couple. I think one is a lot more important than the other. So I'll talk about the unimportant case first. So the unimportant case, what you could have a blind spot because combinations of those features simply don't co-occur. Like there's no data in that region because perhaps it's, depending on what you're measuring, it's physiologically impossible to generate data points in that region. And so you have a blind spot. In that case, it's not very problematic. If you have enough data in the other regions, then it really doesn't, the blind spot is not consequential. I think where it becomes a lot more consequential, and what I'll sort of propose is more the standard, is when you have a training data set for building a model that's really biased in an important way. There's a paper that recently was published that stated that they did sort of a meta-analysis of machine learning models in the clinical AI space, and they found that 70% of the training data in clinical AI models comes from 3 states: California, Massachusetts, and New York. And so, I mean, to me, that seems like a massive problem. But so what happens when you have data from 47 other states or outside of the US, right? And the question is that we don't know, because you effectively have a blind spot in that region of data. So in cases where you have a data set that's biased in some important way, then the blind spot comes out because you're effectively undersampled, you don't have a diverse enough sample, and so on. And in this slide, I actually have this really simple example that shows why this becomes a problem when you deploy a model. This is the same 2-dimensional problem from the previous slide. And let's assume that I've given this problem to 2 different algorithm designers. The first one has designed a classifier that has a decision boundary that is shown on the left, and the second one has designed a classifier that has a decision boundary like the one that's shown on the right. And all this means is that, let's say all of the samples in this region of feature space belong to the MCI class according to this example, and all this, for the second algorithm designer, all the samples in this region of feature space belong to the MCI class, and the other sort of half space belongs to the other group. If you sort of follow best practices, both of these classifiers exhibit approximately similar performance if you were to measure it using cross-validation or on the training set. What I want to call out is the fact that they make very different decisions from data that was to come from the data set blind spot. So one would call them healthy, the other would call them as cognitively impaired. And so as you're building the model, you wouldn't know this because you have no regions, no data from these regions. So both models work equally well. However, let's say you deploy a model and now you deploy it in one of these 47 other states and you get data from this blind spot and you end up making a call. You wouldn't be able to pick up that there's a problem even until after the model was actually deployed and making decisions in the real world. Okay. So what evidence exists that this is actually a problem in the published clinical AI literature? We can't get a direct evidence for this, right? Because most of the models that are published in the literature aren't deployed to see if there's a mismatch in the performance reported in the paper versus deployed. But we can get some indirect evidence. The indirect evidence comes in the form of a sort of a simple fact about models, right? If you have a really well-trained model, as you add additional training data, the model performance should either remain the same or should increase if this data contributes to additional insight the model didn't have previously. However, we did this meta-analysis of 52 classifiers published in the literature, about half of them looking at the problem of healthy controls versus patients with Alzheimer's disease, or the other half looking at healthy controls versus patients with other forms of cognitive impairment. What you notice is this negative trend, very unexpected. So as the sample size is increased, the reported accuracy in these papers declines as a function of sample size. And this is actually observed not just in speech, as we reported in this model, but in neuroimaging studies, in eye tracking studies, across different diseases, not just Alzheimer's disease, but schizophrenia and depression and so on. So any clinical AI models that used high dimensional data, but where the model developers were sample starved, so to speak. And so this negative trend we interpret as basically that there's a over-optimistic sort of estimation or reporting of accuracy in the literature. It's very likely that I think some of these low sample size studies are overestimating the accuracy of the models should they be deployed. And the reason for this, I mean, we argue that there's three reasons for this. I'll sort of very briefly touch on them, and then we can have a discussion if people are interested. The first is some form of overfitting. Let's say the algorithm designers didn't follow best practices. Perhaps they used a training set to do feature selection or something like that. I think that's less likely, because you'd expect, I think most individuals would follow sort of best practices when it comes to supervised learning. The second variant of the same problem is where you have a group of people banging away at the same data set using best practices, but over and over again over time. So, changing the hyperparameters of their model, or figuring out edge cases where their model is failing and sort of patching them up. And there you're overfitting to the data that you have using sort of not traditional, the traditional definition of overfitting, but manual overfitting, if you will. And then the third, which I think contributes heavily to this, is that these models, the reports of accuracy are themselves random variables and are themselves variable. However, the only ones that are published are the well-performing models. For every model that reports performance of 90%, there's probably one that has a performance of 45%. It just never gets published. And so there's some sort of file drawer effect that's perhaps happening. These are the three different reasons for this, but the blind spot contributes to each of these explanations as we outline in the paper. Okay, so when we were writing this paper, it was like through a series of virtual happy hours with my co-authors, and it'd get kind of depressing at times because we talk about all the different ways in which models fail. And so we had a section where we talked about, well, what can you do to mitigate some of these issues? And actually writing the section of the paper was kind of cathartic, talking about the different ways in which you can build a robust model. Is provided some psychological release. So I won't go into the details. That section I think is probably most in depth. We followed the FDA's total product lifecycle workflow and provided recommendations for each box in that block diagram. And broadly speaking, they correspond to these bullets, although I think there's a lot of detail, important detail behind each one. I'll touch on these for a minute. The first is that you want to reduce the dimensionality of the data generating process. And stated in simple terms, that means people talk about passive versus active sensing when it comes to clinical models. It's really, it's obvious why people would want something that sort of, Alexa to listen to a person speak and then make a determination about whether they're healthy or whether they have some form of cognitive impairment. But fundamentally, that problem has too much variability embedded into it. It's a super high dimensional problem. A more appropriate instantiation of that problem that can help you build a more robust model is to have what we call tasks, speech elicitation tasks that are taxing in some way, where you're reducing variability and asking the individual to do something that's difficult and that might highlight different forms of cognitive impairment. The second is to collect large and representative training data. This is easy to say, really difficult to do, because by virtue of us using digital sensors, these sensors are sensitive to everything. So if you're using, say, an actigraph, and you want to use the raw data from an actigraph, that's going to be, it's going to correlate with how big your house is if you have to walk around your house, right? Like, that's obviously not a clinically important parameter. So you want to make sure that you sort of represent the training set appropriately. The third is that to use features relevant for the task at hand. And again, this is easy to say, very difficult to do. I think in terms of how you go about doing this, very early on, if this is a brand new problem, I think it makes a lot of sense to spend a lot of time injecting domain expertise into the solution. So doing feature engineering, perhaps. As you acquire more and more data, there's all forms of transfer learning that can be done that we talk about in the paper. And then the final one, and I'll end with this, and then we can open it up to questions, is this idea of distribution shift post-deployment. If you train your model on data from 3 states and then you deploy it and it's being deployed outside of those 3 states, it's perhaps the case that there's going to be a change in the distribution of the data. You're now going to be filling in those blind spots with samples from regions that you have previously not observed. And there are ways to check for this. There's ways to check to see if the data that you're getting post-deployment is aligned with the training data used to train the model. But I thought this was a really forward-looking thing by the FDA. They have a mechanism in place by which to recalibrate the model should you determine that there's been some sort of change in the performance of the model, or if you want to improve the model. So we make some recommendations about what other parameters to search for or to check for post-deployment to ensure that there's that the model isn't performing in some unusual or unexpected way. So I'm gonna pause there. I can talk a lot more about this, but I'll pause there, and hopefully that generated enough interest for some questions.
Visar, that's absolutely terrific. A really phenomenal overview of an important piece of work. I'll invite folks on the line, and we can probably stop sharing and invite folks to put their videos on if they are somewhere where they can participate in the conversation. Definitely feel free to drop questions into the chat or raise your hand, and we can make sure that you have the opportunity to pose your question. Julie, if you are somewhere where you can unmute yourself for a moment, Visar gave you a really nice shout out at the beginning as being a sort of partner in all of this. Sort of any commentary that you would like to add, any additional reactions to the phenomenal sort of overview Visar provided this morning?
Yes, absolutely. Let me just get my video on here.
I'm sorry, I sprung that one on you horribly. Thanks, Julie, for being a good sport.
Yeah, no, this is, it's been a wonderful journey that I've been on with Visar, and from where it all started to where we're going. And in my previous work, this was before we could do a lot of automated analysis. We were restricted to measuring aspects of the speech signal that we could do essentially by hand, right? Or with minor automation. And it was obvious for many, many years that there are aspects of the acoustic signal or aspects of language that are highly indicative of certain types of neurological degeneration. And so it was just amazing to be able to get to the point where we were able to use the kind of the knowledge that science has given us about those aspects, and then move it into widespread applicability. And then this promise of AI that I think Visar probably, I think I came in right as you were talking about our failure. We were so excited about the promise of AI in the early days, and then just kind of finding the ability to make this work and to make it scalable has been just magnificent. So I'm happy to take any clinical questions as well.
Fantastic. While I continue to watch the chat and sort of the participant list for raised hands, I have a couple of questions if I can, and forgive me both. I am many things, as I'm sure the team will tell you here at Dime. I'm not a data scientist. So, but one of the things that I find really interesting is this idea of sort of data blind spots. And it feels sort of interesting as someone who has sort of monitored and been in healthcare research for sort of decades. And there's been this, we've had these tiny snapshots of data, right? We've had these sort of episodic data points, and it's hard to sort of fathom that we are still sort of talking about such big blind spots as we enter this high dimensionality, as you sort of appropriately describe it. I was wondering if you could explain, just so I have a better grasp of it, tell me, even though we have much more data, how these blind spots can be so damaging. Because instinctively, I sort of feel like, well, surely it's better than it was before. Is it because AI always finds a relationship? Like, tell me more about, like,
yeah.
Help me unpack this a bit.
That's such a good question. That's such a good question, right? So how can you have higher volume blind spots when you have so much more data than you had before? Yeah, it's such a good question. So previously, I think we were, especially in neurology, when you were assessing how well a patient was doing, you were looking at something like a functional rating scale, right? And perhaps that they were evaluated at some sparse interval. And so these individually, each of these instruments were validated for reliability, for repeatability, for clinical relevance. And so if you noticed change, you could be sure that the change was a result of some change in the clinical condition, right? You could be relatively sure. So now enter thousands of sensors. Like every single one of us has at least 5 or 6 sensors on us at any given point in time, right? And if you're talking about tracking that data longitudinally and trying to extract clinical meaning from it, that's a really challenging problem. I mean, it's like just because these parameters change over time, it doesn't mean that they're changing for some clinically important reason. I mean, they could be changing because one day you decided to take a different route to work or whatever, right? I mean, nothing to do with your clinical condition. So what's really challenging is trying to apply a data-driven approach to extract clinical meaning from these high dimensional signals. In the defense community, I've heard it called as the problem of not finding a needle in a haystack, but finding a needle in a needle stack. Everything looks the same. So finding the right needle that is clinically important for the question at hand has become much, much harder. Previously, that was done by construction. Like we constructed these instruments so that they were clinically relevant. And hopefully that answers your question.
It does. It does. It's really, it's really good actually, which is, while those data points were snapshots, we were much more confident in advance that they were indeed the right data to be monitoring. And because they were, for want of a better expression, a bit of a blunt instrument, if we really were seeing sort of clinically meaningful change in those, it was highly likely that that was indeed a sort of correct signal. And I like the right needle in a needle stack. It's a good visual that helps here. I want to keep sort of pushing on this a little bit here as I think about what do we do about this, right? I want to go to the bit of your virtual happy hours, the happy part of it, and we think about this sort of going forward, right? How do we think, who's responsible for doing this testing and evaluation, right? It's all over the sort of tech press right now. There's always a sort of, controversial and very well-written piece about sort of bias and misses and all these sorts of things, but who's responsible? And if no one, what's the system we should create to get there?
Right. Yeah. So I think responsibility is probably spread over multiple levels. And I actually just to give a shout out to Dime, because the V3 framework that you guys have put forward is, I think, the very first step in this. It provided a terminology for working with biomets and digital biomarkers that I think is absolutely critical. So, and a framework for how to go about doing this sort of validation. In terms of who's responsible, I think there's some obviously individual responsibility associated with the different entities building these models. I mean, I like to remind folks that this isn't like speech recognition, where if Alexa doesn't understand what you're saying, I mean, who cares? You just say it again. So you say it slower, you say it with, you know, over articulation until she does understand. You know, we don't have the benefit of that here. I think the stakes are much higher. So developing clinical grade analytics is level and above higher in terms of complexity than building consumer grade products. And so that has to be built into any organization that's developing these models for deployment. And the second is obviously a top-down approach, right? Where I think the FDA has done a really good job of providing guidelines. You know, sometimes, you know, we wish they would happen faster, but it takes time to do these things. And so they've done a really good job of providing guidelines for how to go about developing initial versions of the product. And then what I really like is this idea of continuous monitoring of the model. So even post-deployment, by virtue of you introducing a new clinical AI tool in the workflow, that may change the way that you know, the types of patients that might come in clinic, and which may change the distribution of data that the model is observing, which means that you have to recalibrate. So I think that there's sort of a bottom up and a top down responsibilities here.
I love that response. And one of the other things I think FDA has done really nicely, and sort of we've attended some of them, I know a lot of members of the DIME community have, is the Patient and Family Advisory Council has also been sort of listening carefully and sort of organizing these listening sessions to really understand, look, what are the risks and considerations sort of vis-a-vis these sort of AI approaches that we're starting to deploy, and how do patients think about this? Julie, I want to sort of pitch that over to you if I can. I mean, I think about the conversations I have all day, every day. Like, what should we be saying to our clinical colleagues right now? How trustworthy are these things? How ready for prime time are they if you don't live in Massachusetts, California, or New York? And as a Florida resident, I can tell you we are definitely different.
Yeah, I think that that caution has to be the name of the game, and you know, when clinicians are you know, using this as a source of information, they need to be offsetting it with their other clinical tools and you know, their tried and true best practices, and you know, using it to augment, but certainly not to replace. You know, this is you know, a serious problem. And as Visar said, you know, the consequences and the risks are very high. So at this point, I just extreme caution.
It's one of those things where I sort of sigh, and then I agree with you. We want to say, gosh, these capabilities are here. We're able to start sort of making these connections. But I think, Visar, one of the points you made that was really powerful was around, you know, this isn't, and I'm adding on top because I have a habit of doing this, is, you know, this is MCI, right? This is potentially someone looking at, gosh, do I need to go back, and do I need to go and start sort of looking at whether I'm at risk of Alzheimer's? There are sort of serious consequences with seeing a signal in this data and receiving this information. This is not, gosh, your banner ads might not be for the right pair of sneakers. So, you know, I think this is important, right? Sort of tempering our enthusiasm and the capabilities of this technology with, you know, not only the caution that you mentioned, Julie, but like a whole new set of harms that we have to be eyes wide open to. I have a couple of questions, but starting to get some from the chat. Nina, are you somewhere where you can unmute yourself and pose your question live?
Okay, what's the...
No problem. All right, Nina, I will give, okay, not at the moment. I'm sorry, I didn't mean to put you on the spot. Let me give you voice to your question. So really good one here. request from Nina to see if you could please talk a little bit more about the difficulty from going to the lab to the clinic, that sort of translation of these models. What barriers did you identify that limited the model's external validity? And have you, and how, I added that, have you been able to mitigate some of those barriers? Great question, Nina.
Yeah, this is actually an excellent question. So in terms of the work related to speech, I can speak to that. I'm sure that there's other sort of idiosyncratic issues associated with different other modalities. The problems, there's some fundamental problems in speech analytics that have yet to be solved. And I'll highlight a couple that might seem trivial, but they're actually not. So imagine you have two people speaking on the same microphone. So being able to pick out when one person starts and the other stops is really, really difficult, especially if they sound similar. And so in the lab, you don't have problems like that. You have complete control over the means by which you collect the data. You have complete control over when the participant begins to talk and when they stop talking, because you have a coordinator there. So you can really make sure that the data is of super high fidelity. When you deploy this in the real world and you're scaling this to studies with thousands of participants, and they're doing this from the comfort of their own homes, that's no longer the case. So being able to sort of develop technology around ensuring that the data is of sufficiently high fidelity. And that means, designing a user interface that allows for that, identifying when there might be contamination in the data and perhaps either requesting new data or removing it altogether from the analysis. Those are the sets of tools that you have to build around sort of what I'll call research grade analysis tools in order to get robustness post-deployment. The other I'll mention, and it goes to what Jen mentioned earlier, which is this idea of actually engaging with patients early on as you're developing this tool. I think that's really critical. I mean, one of the first studies we did this was with individuals with motor impairment. And everything from the size of the text on the screen to the size of the buttons to make sure that they could be sufficiently pressed, and to how sensitive the screen was in order to be able to press the buttons. Those are all things that, as a scientist in the lab, you perhaps don't pay a whole lot of attention to, unless you're actually studying those. But if you want to deploy this in the real world, you absolutely have to. So is there a lot of communication with patients and users of this technology. Julie, would you add anything to that?
Yeah, I would say that the other piece is that we are not using a purely data-driven approach. We are focusing on speech metrics that we know from previous research change in certain neurological conditions. And these metrics are validated using the V3 method. And we are certain that when we measure these things and they change over time, that they're reflecting changes in the clinical condition. So that's another way in which we increase the external validity.
Sorry, I was going to say fantastic. These are the important conversations, right? When we want to go from sort of what's possible to sort of actually manifesting these technologies in such a way that they help sort of clinicians and the patients we're all here to serve. These are the sorts of conversations we need to have. And I'm grinning ear to ear that V3 keeps coming up. Kelsey, you also have a really good question here. Are you somewhere where you can unmute? Perfect. Go for it.
Yeah. Hi, thanks so much for the great presentation. My name is Kelsey Taka. I work at W.O. Gore and Associates, and really appreciated the comment around the data that is being collected may not have all of the information needed to make these clinical conclusions. So you gave the example of like changing your routine of walking to work being not at all related to your health outcome. So could you speak at all to maybe gaps that you see, or the biggest gaps between wellness products that are being used in this space versus those that are clinical grade, or maybe they don't even exist yet and there's kind of a goal that we're trying to reach as it relates to this data quality, and maybe some of the biggest gaps, areas to focus when we're developing these new products?
Yeah, great question. So I guess I can't speak specifically to wellness products. I don't know that domain very well. I mean, the sort of secondhand knowledge I know is that there's they're able to escape some of the regulatory rigor that health products can't, right? And I view that as potentially consequential if individuals are making decisions about their lifestyle based on the outputs of these apps. But that aside, I'll talk about the second part of your question, which is what are the gaps that exist? I think that there's a tendency, and I understand it because it's really convenient to try and short circuit the scientific method, where if we just collect enough data, then the data will tell us everything we need to know. And I think high volume data is really important, but I think collecting the right kind of data, investing in experimental design, understanding how the different digital endpoints that we're measuring in different studies, for example, how they vary by different strata in the population, young versus old, different demographic groups, high SES versus low SES. I think that's really critical. And so what I wish would happen would be some sort of large scale study. I'm talking N equals hundreds of thousands, where we just understand how some of these parameters, these things that we're measuring, vary by different strata in the general population, not necessarily focusing on a clinical population per se. I think there's actually groups that are working on these sorts of studies, but I think that would be massively beneficial. The second is having really targeted solutions to specific problems. So I'll use Dime as an example because I think they've done this well with their the recent collaborations around, I believe it's sleep, right? Yeah. And so getting stakeholders together and coming up with solutions to perhaps narrow but important problems. Because just like with anything else, I mean, if you propose a solution that is a panacea for everything, it likely doesn't solve anything. So I think there has to be a lot of targeted efforts to understanding specific problems, but then also a large effort to understand how these things vary in the general population.
Thank you so much. Yeah, I think it definitely addressed my question. It sounds like more around the validation than on any specifics as it relates to the product, right? The sensors themselves or the data that's being collected, or, the positioning of the sensors. Like, it's more about the validation evidence to support the conclusions being made. So thank you.
Yeah, I think, sorry.
Ahead, Lisa.
Yeah, I was going to say all of that play a role, right? I mean, the way I think about this is that we have a lot more degrees of freedom than we did before. I mean, you just mentioned a few of them, like where do you place the sensor? What sensor do you use? By virtue of having more degrees of freedom means that your search space is much broader and much larger. And it makes the problem just in general more complicated. That's why you have, 45 solutions for measuring gait. And so then, like, if you want to measure gait in a study, the question becomes, which one do you use? And it's like, I'm not sure. So I think hopefully with the efforts that Dime is supporting, that some of these questions will iteratively over time get answered.
And I want to riff off this, and Julie, Visar, I'm happy to be sort of proven wrong here. Kelsey, I really like your question around the what about the sort of the commercial tools vis-a-vis the sort of medical devices. I think this is really important. And one of the things when we did V3 was we didn't say, look, this is narrowly sort of circumscribed for medical devices. I think there are opportunities for commercial products to do a really good job of this and tell stories in plain language about what they did and how they were inclusive. And there is an appetite for this. And people are asking these questions as they're trying to go through the 300 something health apps and figure out what's sort of safe and what's effective and what has evidence. There's not a mandate and there isn't an oversight, as Visar, you described really nicely. I also think, and this is where perhaps it gets a little bit more controversial, which is the medical devices are sort of regulated, I think, in a very thoughtful way. They're starting to think about sort of really strong post-market activity. I think there's also a lot of assumptions made around the what it means to be a medical device. And Kelsey, you just gave some really good examples, right? If you take a really well-validated, high-quality product and you deploy it in a way in which it wasn't tested, or in which it wasn't, it's not the intended use, it's an irrelevant data point at that sort of at that juncture. So I think it's an interesting tension, but I do think there's a lot of opportunity for anyone, whether in the medical device space or on the more sort of commercial side, to really sort of dive in and differentiate themselves by taking the time to do this well, to collect that data, right? To take the time to or maybe accept a slightly less goodness of fit by not doing gymnastics on the edge cases, right? I think there's a lot of opportunity.
And I think building off of what Visar said, like focusing on a specific area and really proving it out there, rather than kind of this jack of all trades kind of thing where your device can do everything and then it does nothing well. So I really appreciate that comment.
And Visar, as you said, that's what we're thinking about at Dime, right? How can we have these tight problems and then think about translation, as opposed to how can we try and boil the ocean and get exactly nowhere? We've got a lot of questions to get through, so I want to make sure we get to them. Smit, let's come to you next. I thought yours was an important question, and thank you, Kelsey.
Fantastic discussion, Visar and Julie. Really like it. One of the things, as we are thinking about algorithms, and I took it from the discussion that, if the training data, the collected data before we start the algorithm chain is probably not diverse enough, or is lacking the diversity, equity, and inclusion criteria, it may creep up the chain and probably through the algorithm create this bias. I was curious around the fact of like the disparate impact that in the AI world people discuss. How can we, or what are some of the measures we can think about from the standpoint that will help support, either it's from the starting point versus either it's once the algorithm is being developed or it has already been developed and we want to redesign that. I'm not a data scientist, but I wanted to just include the fact that how we can make it more DE&I sensitive so that we capture the data and proposition for all population and not common.
Yeah, very, very good question. So look, I'll talk about the status quo first, and then I'll talk about how I wish it was. So I think the status quo is, hey, I want to build an algorithm. What data exists? Oh, this data exists. Let's use this data, right? And so then by doing that, you run into the situation where most of the data that exists comes from a very sparse set of hospitals around the US perhaps, and so you get this biased sample. How I wish it was is you say, I want to develop an algorithm. Great, what data do I need for that? Well, what sensor am I going to use? I see, I'm going to use a microphone. What are all the different parameters that a person's speech is impacted by? Well, impacted by dialect and age and gender and demographics and a whole host of different parameters. So then you say, okay, well, then what are the strata that I need to sample in order to get a representative data set, right? And so then you do that, or you subsample from a large cohort a set of samples that's representative. Now, you don't know what you don't know, so there could be other parameters, ones that you haven't sampled, that after you develop the model, you find out are important through this continuous monitoring phase. And at that point, you can look at mismatches between the deployment data versus the training data to see if there's blind spots that came about because you didn't think about them during the experimental designer and so on. So that's the way I wish things were. I think they'll get there. I mean, in the grand scheme of things, we're kind of in the early stages. There's like doing science is hard and it takes time. And like, I don't care how many algorithms you throw at things, like it takes iterations. Iterations take time. So each iteration perhaps is a little faster now than it was 15, 20 years ago, but the number of iterations, I'm not sure if that's changed. So hopefully that answers your question. Julie, I don't know if you want to add anything to that.
Only that that if you don't have that control, the relevant variables controlled, particularly the DE and I variables, you run the risk of building models that either artificially assign disease or degeneration to certain elements of the population, or you have them neglected. So it's very risky if those things aren't taken into consideration.
Thank you. Really appreciate that. And I love the shout out on what good looks like and the fact that we don't have to think about it once we start, but think about what we need before we start, and how those parameters impact. So really appreciate it.
Terrific questions, Matt. Visar, Julie, we're looking at some of the incentives to support that more deliberate approach in our data CC. So we might be doing some follow-up to think about what those incentives should be as we go forward. But I don't want to take away from the tremendous questions that we have left as we get to the top of the hour. Yayan, your one piles on really nicely to this idea of sort of controlled data. Do you want to unmute yourself and ask live?
Yeah, hi guys. Yeah, I was really, again, I just wanted to build on Kelsey's question around, moving, into the real world. And I think, as you mentioned already, there are positives to that, in terms of the volume of data. And I think perhaps it maybe gets us closer to behaviors and away from, aspects of performance, right? With all the variation that comes with that, perhaps giving us a slightly better understanding of the daily lived experience or fluctuations in symptoms. But I was just wondering how you're sort of thinking about approaching that challenge and sort of absorbing that additional contextual information and, kind of how you're weighing up the pros and cons of that step.
Yeah, very good question. So I think what I'll say is an opinion, and then I'd be curious to see if Julie agrees with me or not. This process is iterative. I think early on, and as I mentioned, I still view that this is us as a body being early on in this journey. It's important to control different variables that are controllable so that you can understand, for example, the impact of a particular clinical condition or a change in some clinical parameter on the thing that you're measuring. And to the extent that you can reduce nuisance in the background, whether that's because you're collecting it in clinic or you're using the same type of microphone or whatever, right? I think that's a good thing. One, and sort of that's, version 1. Version 2, you can go to allowing some of these nuisance parameters into perhaps a more natural data collection process. And the reason you can do that is because you've learned something during version 1. You've learned something during iteration 1. And so I think to get to the point where, I'm assuming by at home here, you're referring to sort of a passive collection from a microphone that's sitting on your table at home. I think we're probably 5 to 10 years away from being able to say something meaningful with data like that. I think we're still at the point where you have to impose some control over what you're collecting and how you're collecting it. Now, that control has loosened up. I mean, even internally in the sort of work that we do, initially, everyone used the same device and everyone collected it exactly the same. Now we allow for different devices and we allow for some variability in microphones and the types of speech samples that people provide. So that this will, get better over time. But I think relatively speaking, we're still early stages.
Yeah, I would just add, it really depends on the use case as well. So if you're trying to identify, for example, early onset of detection of disease, you're going to ask people to do things that they normally wouldn't do, something that's more taxing, more challenging, in order to kind of put a magnifying glass on their capabilities. You might be able to get at something similar to that by following someone in their home, noticing they don't talk as much, noticing some things that are a little more coarse, So it's really kind of the use case. So I can see there might be some benefit earlier on before we get to the point where we have valuable passive data collection, just in terms of the warning light, that something's going on here. But in terms of really identifying something in the early stages, I think taxing situations are important.
I love those comments, Julie. And I also think it's made me think of, we also need to be good stewards of how we interpret this information, right? We might see a signal that might lead to some sort of intentional watching or some follow-up. It doesn't have to mean diagnosis, right, based on this. And I think actually that's a really important point here. We've got just a couple minutes left and a couple questions. So let's see if we can go quick fire. Shira, are you somewhere where you can unmute and ask your question live?
Oh, hey. Yes, I am, and hopefully I'm not echoing on Visar's computer. I'm pretty close to where he is. Okay, great. So my question was, a theme of today's really interesting discussion has been blind spots, and there's an uncharitable view where no matter how well intentioned you are and how much testing you do, when you have high dimensional data, you can never sample the entire space. In the end, you must make a decision and deploy. And so I wanted to raise as a discussion point for this group in particular, who's, especially, Jennifer, you made the comment earlier about who should be in charge of the testing and what kinds of safeguards need to be in place. I wondered if there's a need for an intermediate sort of device where, listen, we followed the V3 framework, we did the best we can to do some testing and made an honest attempt. But in the end, we don't know if this is going to detect really early cognitive impairment until we get a bunch of people to try it, right? So is there like a place in the world for a kind of device where it's under that kind of testing, but still a lot of scrutiny, kind of like an early stage device? And how might we go about implementing that?
That's pretty interesting. So that's almost like, in our instantiation of the problem, we assumed that the hardware was fixed and that we're sort of building algorithms on top of it. If I understood it correctly, you're suggesting that there's perhaps a way where you could actually jointly optimize the hardware with the software, where you have some intermediate device that has some flexibility in perhaps what the microphone looks like, or maybe where it's positioned or something. And that's, yeah, I mean, I haven't thought about that, but that's really intriguing.
So certainly can't speak for our VA colleagues, but we have the privilege of a partnership with VA here at DIME. And one of the things that they've been actively thinking about as the largest integrated healthcare system in the country is, can they be a partner for the development of these sorts of things? Because if you think about, it's a national reach, it's a very diverse population of former warfighters who now seek their care at VA. And actually, whether they move forward with this or not, and again, I certainly don't want to sort of speak on their behalf, but conversations we've been having with them, I think they're being very innovative in considering, do we want to wait for a fully baked product, or do we actually want to, as part of our public health mission and as part of potentially bringing high quality tools that serve all of our veterans to bear sooner, could we go earlier with a tool that has sort of reached X threshold, and working together to get it to Y? And so I actually think that's a really interesting opportunity there. And Shira, I saw you smiling. I'm not sure if you have any reactions there.
Oh yeah, it's like a teaching hospital for devices. I think it's really a cool idea. Yeah.
Well, and even, they have a nice relationship with FDA as well. So is there even a way where we can start to really fully sort of connect the dots here? And I think that's part of a really sort of bold vision that our VA colleagues have. So lots of opportunity there. And I think a really good question. I was multitasking. I was grabbing the link to the DIME Slack team. There's still some really good questions in the chat here. Visar, with an able assist from Julie, this was great. You guys wrote a phenomenal paper. It is really important, I think, that we all grapple with this. And even if we aren't experts doing the hard data science, recognizing we're sort of early, we all have to be sort of discerning consumers of these tools, these products, this information, and be good stewards of the field. This was absolutely fantastic. Terrific conversation, everyone. Thank you so much for all of you who stayed on the line, posed terrific questions. Visar, Julie, thank you for a terrific contribution to the field. Thank you for taking some time with us today. Before we log off, I'm going to share just one more thing before we break. For folks who enjoy our conversations here at DIME, our next journal club is going to be discussing another well-titled paper, listen up, using digital tools when designing patient-centric clinical trials. So everyone on the line, thank you so much. Visar, Julie, appreciate your efforts. Thanks for spending time with us. Have a great day, everyone.
Thanks, everyone. If there's any questions that we didn't get to, please post them on Slack and we'll follow up.
Appreciate that.
Thanks so much. Thank you. This was very interesting.
Terrific. Thanks. Bye-bye.